Hello everyone, welcome to the St. Joseph's open web-based learning platform, that is how you learn. Also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level.
Welcome back to the Plant and Animals Biotechnology course. This is a module 5, Bioethics, Biosafety and IPR, Intellectual Property Rights. Bioethics in cloning and stem cell research, biosafety, GLP and GMP. Intellectual property rights are the topics in the module 5. Will be discussed by Dr. A. Veerabrahman, HOD, Biotechnology, St. Joseph College for Women, Autonomous, Visakhapatnam. So these are the learning outcomes for the bioethics in cloning and stem cell research. Acquire the knowledge about the bioethics. Propose and implement these scalable protocols for biosafety. Get an acquaintance and GLP, good for laboratory practice and good for manufacturing of the products for the society, benefit of the society. And be proficient, students will be proficient in generation of intellectual property rights. These are the contents: bioethics in cloning and stem cell research, biosafety, good for laboratory practice and good for manufacturing, patents, trademarks and copyright, personalized skills, personalized protocols, marketable, scalable, commercial aspects. Let us discuss first the first topic, bioethics in cloning and stem cell research. Bioethics, biosafety and IPR are crucial considerations in biotechnology, particularly concerning cloning and stem cell research. Bioethics focus on the moral implications of scientific advancements such as cloning as replica production and stem cell research, prompting debates about human dignity, potential for harm, and access to treatment. Biosafety ensures the safe handling and containment of biological agents, including genetically modified organisms, to protect human and environmental health. GLP or GMP guidelines are vital for maintaining the quality, reliability, and integrity of research and manufacturing process. Intellectual property, including patents, provides legal protection for inventions, personalized inventions or group inventions or college inventions, organizational inventions, which can incentivize innovation but may also raise concerns about access and affordability of treatments. The first topic, bioethics in cloning and stem cell research. Cloning raises ethical questions about the potential for reproductive cloning, the creation of identical individuals, and the impact on human identity and individuality. So this is somehow an Indian scenario. This is a very ethical issue. Stem cell research involves ethical dilemmas related to the use of embryonic stem cells, including concerns about the moral status of embryos and potential for harm. Nowadays, the public, of course, couples are very interested to get their stem cells from the umbilical cord after the gestation, after the pregnancy, delivery. They are asked to preserve the cord blood, okay, which comprising most of the concentration of the stem cells. Genetic engineering, of course, union of the two different source of DNA molecules prepared as an engineered molecule raises concerns about the unintended consequences such as off-target mutations, maybe how the molecule can deviate, can modify or design, and the potential of misuse of genetic technologies. This is very important. Everybody must know about the consequences of pros and cons about the functionality. And this is the schematic representation for two kinds of human cloning. Isolate the somatic cells, person, isolate somatic nuclei, egg, anucleated egg, nuclear transfer, activation shock, early embryonic development in the laboratory, then host mom, surrogated moms, representative, clone the baby, therapeutic, of course in the laboratory, embryonic stem cells are cultured, whether their potentiality, how they are maintained. So this is the world views about the bioethics in cloning, stem cell research in the Australia has prohibited human cloning. Therapeutic cloning is now legal in some parts of Australia. As per Canada, prohibits the following cloning of humans, cloning stem cells, growing human embryos for research purpose, and buying or selling of embryos, sperm, eggs or other human reproductive material. India, our country, doesn't have specific law regarding cloning, but has guidelines prohibiting whole human cloning or reproductive cloning. India allows therapeutic cloning and the use of embryonic stem cells for research purpose only. As of now, it has been prompted, it's been resisted. In Argentina, prohibits experiments concerning cloning of human cells in order to generate human beings. So this is of course, in a trial and error method in animals. Animal cloning has been already obtained in 1960. Jan Wilmot produced the dolly. After that, has been died. No scientist has been prepared the animal clones. So these are some ethical issues of human cloning: history, process, religious aspects, federal and governmental regulations, possibilities and consequences, world views, medical issues especially. The next topic is biosafety containment zones, GLP and GMP. BSLs are a system of biocontainment precautions used to isolate dangerous biological agents in a biolaboratory setting. They range from BSL 1 to BSL 4, most stringent, least stringent BSL 1, with each level requiring specific safety practices, equipment and facility design. Containment, focusing on preventing the release of harmful organisms into the environment, protecting human and animal health. So containment zones already we observed in case of the last corona situation. Every village, every street has been a containment zone if the virus is outburst, right? So the biosafety cabinets are used not only in the laboratory. Once the epidemics or become a pandemic in a geographical region or an area, there will be containment zones. GLP ensures the reliability and validity of research data by establishing standards for laboratory operations, including quality assurance, record keeping, and study conduct. So this is a very, very important aspect, GLP guidelines. Each pharmaceutical industry have their either a IP or USP or a BP or a GP. Any based on their practice, the laboratory must fulfill the criteria to produce the standard drugs. GMP establishes standard for the manufacturing of pharmaceuticals and other products, ensuring quality, safety and efficacy. Okay, so pharmaceutical drugs which are treating a variety of diseases in human beings must be produced in a variety of protocols and released into the atmosphere. So these are the BSL-1 microorganisms through observed microscopic animals, plant, arthropods, insects, aquatic organisms. Biosafety level 2 facility designation animal biosafety level, plant biosafety level, insect biosafety level, aquatic biosafety. So each of the levels, organisms have their own. In that level only they can be concerned, they can be dealt. In BSL 3 and 4, 1 to 4 are animal biosafety level, 1 to 4, like that. World Health Organization designs laboratory facilities under 4 levels: 1, 2, 3, 4. Right? So level 1, level 2, level 3, level 4. Basic teaching and research, GMT, and safety equipment, there is nothing. Simple detergents or surface agents, open bench work. Biosafety 2, primary health services, diagnostic services, research. Practice, good for manufacturing trained skills plus protective clothing. Biohazard science must be informed to the patients and as well as the students and workers. Open bench plus biosafety cabinet for potential aerosols. Safety level 3, specific diagnostic services, research, especially focusing on R&D, research and development. Special clothing, controlled access, directional airflow. Biosafety cabinet, other primary devices for all activities. Safety level 4, dangerous pathogen units. So these zones are completely away from the earlier 3. 3 plus airlock entry, shower exit, special waste disposal. Safety cabinet, positive pressure suits in conjunction with the class 3, class 2 cabinets, double-ended autoclave to wall filtered air. The next topic in the chapter is patents, trademarks, copyright. Gene patents can restrict access to genetic information and limit research, potentially hindering the development of new treatments. So patent is a personalized innovation, so it must be protected through a protocol designed by a country, designed by an agency that is mandated by the government. Incentivizes innovation by providing intellectual property protection, but can also create barriers to access, affordability of treatments, ethical considerations. Informed consent, ensuring that individuals are fully informed about the risks and benefits of research before participating is essential. So one must be completely aware about the risk associated with the program, the patent, the protocol, so that the person who owns the patent as usually follows the risk and assess. Ensuring that all individuals have access to beneficial biotechnological advancements is a crucial ethical consideration. Everyone, public awareness and dialogue, open and transport discussions about the ethical and societal implications of biotechnology are necessary for informed decision making. This is very important. So let us discuss the copyright, trademark and patent. Copyright established act was established in 1957, protecting original creative expressions like literary works, artistic works, dramatic works, valid for lifetime of the author plus 60 years after his or death. Protection available in most of the countries in the world. Copyright sources creative of intellectual creations. Right comes into existence. Exclusive rights over the copyrights are created the moment the authorship creates the work. No provisional application is required. No symbolic representation to show registration. Trademark Act has been established in 1999. Protection of unique name, that's a trademark. For example, Pepsi, Limca, or Coca-Cola. These are a trademark, brand distinct, super ring, okay, from other, can include names, slogans, logo, shape, color. So the other person, the other company must not imitate or reproduce. That is a punishable, offensible. Maximum of 10 years can be allowed for trademark. Secure the branding under which products and services are sold. Once the trademark gets registered, the applicant of that mark can claim complete right over the said mark. Registration usually will take 12 to 18 months. Registration doesn't include provisional application, but it requires a trademark search. Used when registration is in process. Symbolic. Used when registration is completed. Trademark. Patent. It's a personal. The Act is established in 1970. Protection of inventions that are novel, original, and has industrial utility. 20 years of validity. Patent sources inventions that are useful for the world and the sum of the use. New inventions in the pharmaceutical industry, that is a patent. Registration takes 2 to 3 years in all, but the owner can stop anyone else from claiming right over a particular patent the moment he applies for provisional patent. A provisional application gets you 12 months to time to file a complete specimen and a priority date claim. No symbols or representation shows registration. So this is a diagrammatic representative, wonderful diagram, patent, copyright, trademark. So that says that common thing is here intellectual property. So between the copyright and trademark, logo is logo and emblem. Between the patent and copyright, software issues. So these are the references about the topics discussed in the chapter, in this unit, in this module. Thank you for your attention. Thank you for your listening. So with this, the course Plant and Animal Biotechnology has been completed. All the modules have been finished. Thank you very much.